Everything we do is available freely on bionichaos.com. Please consider supporting me on Patreon, and don't forget to gently click like, subscribe, and comment.
Imagine, if you will, looking deep inside the human brain. Not with a surgeon's touch, though, but with pure data.
Yeah, what if you could actually see the electrical whispers of thought? Or, the intention behind imagining a movement?
It really does sound like something out of science fiction, doesn't it?
It might seem that way, but it's surprisingly within reach. It's just a really intricate process, starting with, very sensitive measurements.
It really is, and that's exactly what we're going to dive into today. Welcome, everyone, to our deep dive into the fascinating world of EEG electroencephalography. Our mission for you today is basically to unpack a whole stack of technical sources that detail this journey, acquiring, processing, and then visualizing real EEG data.
Yeah, and it's not just about listing the steps, right? It's about seeing how you actually transform those raw, sometimes messy brain signals into something meaningful, something you can even visualize in a pretty stunning way.
Okay, so the big question we're tackling is, how do you take these tiny electrical impulses, often buried in noise, and turn them into something you can actually see and, understand?
Exactly. It's a bit like being a data detective, honestly. You're deciphering this kind of secret language. You find surprising things, you hit roadblocks, but then you get these incredibly satisfying moments of discovery.
I love that data detective. It really feels like that sometimes. You're solving puzzles, maybe hitting unexpected walls, but then having those amazing aha moments when it all clicks. So let's start this journey.
Let's do it.
Okay, so our adventure, our quest into the brain's electrical world, it begins like many quests do, hunting for treasure. But this isn't gold, it's digital. We needed raw brain data.
Right. And our search led us to this fantastic public resource, the PhysioNet EEG motor movement imagery dataset. It's like finding a huge digital library just full of brain activity recording.
A library, okay.
Yeah.
And within that library?
We zoomed in on one specific file, S001R01.edf. This file is just one recording from one person doing a specific task involving moving and imagining movement. A really good place to start.
Gotcha. So how did you actually get your hands on it?
Well, we used a pretty standard tool in the Python world, Rejection, for the download. But here's where you get that first little real-world twist.
Ooh, what happened?
The file downloaded as a .zip archive, or at least that's what the system thought it was.
Ah, okay, so what was it actually zipped?
Exactly. It's a common thing, assuming file types. We had to look closer, inspect it, and realize, this is actually an EDF file, European data format.
EDF. And that's important because
it's a standard format for medical time series data. Knowing it's EDF tells you a lot about its structure right away. It's designed for this kind of work, which is, crucial for sharing and comparing results in research.
Makes sense. So you've got the right file, correctly named. What's next? Loading it up.
Yep, time for the first look. We used MNE-Python for this. It's a really powerful open source library specifically built for analyzing EEG and also MEG data magnetoencephalography.
Right, MEG measures the magnetic fields, often used alongside EEG. So what did MNE-Python tell you initially about this file?
Some really cool stats right off the bat. 64 channels.
64, wow. That's like having 64 tiny microphones listening all over the scalp, right?
Pretty much. 64 highly sensitive antenna, picking up those faint brain signals. It gives you incredible spatial detail.
Okay, 64 channels. What else?
They were recording at a sampling frequency of 160 hertz.
So 160 times every second for each of those 64 channels?
Exactly. Capturing the brain's electrical activity really fast. And the whole recording was about 61 seconds long.
Okay, so those numbers, 64 channels, 160 hertz, 61 seconds, they tell you about the richness of the data, the detail.
Absolutely. High temporal resolution from the sampling rate, good spatial resolution for the channels. It's a really high fidelity snapshot of what the brain was doing over that minute. You can capture quick changes and get a better idea of where things are happening.
Okay, so we have this rich, raw data, 64 channels, brain whispers, 61 seconds. But you mentioned raw data is rarely clean, right? It's noisy.
Oh, definitely. Like trying to tune into a faint radio station with lots of static. That's why the next step, preprocessing, is so critical.
Yeah.
Cleaning up the noise.
Right, got to get rid of the static. How do you do that?
Well, one of the first big steps is applying a filter. We use what's called a bandpass filter, specifically letting frequencies between 1 and 40 hertz pass through.
Okay, 1 to 40 hertz. Why that specific range? Is that where the interesting brain stuff happens for, like, motor tasks?
Exactly. That range covers many of the well-known brain rhythms involved in thinking and moving things like alpha waves, beta waves, theta waves. By focusing there, we cut out really slow drifts, maybe from sweat or electrode movement, and also the really fast high-frequency noise, like muscle twitches or electrical interference from lights and computers. It boosts the signal we care about.
Okay, so it's like tuning the radio dial precisely to the station you want. Makes sense. What else is involved in this cleanup?
Well, we also took a really close look at the EDF header information. the file's metadata.
Like reading the label on the specimen jar.
Exactly like that. It tells you vital details, how the data was collected, how the electrodes were referenced, specifics about each channel. You need that context for correct interpretation.
Got it. And did you look beyond just this one EDF file?
We did. To really get the full picture, we needed more context from the broader PhysioNet database. So we brought in another Python tool, the WFDB library.
WFDB. What did that let you do?
It let us explore the whole database programmatically. We could list all the different recordings, see what else was there. And importantly, we downloaded some related files.
Like what?
Like an image file, 64-channel harbor.png. It actually shows you exactly where those 64 electrodes were placed on the person's scalp.
Ah, the map. So you know if a signal's coming from the front or the back of the head.
Precisely. It uses a standard system, the 1005 montage, which means the placement is consistent, allowing us to compare our findings and understand the spatial origin of the activity. It's the blueprint, like you said.
Okay, the map. What else?
We also got a records file, basically just an index, and crucially, a WFDB TAL file.
WFDB cal.
Calibration.
Exactly. Channel calibration information. It ensures the raw voltage readings from each sensor are accurately converted into standard units, like microvolts. Accounts for tiny differences between sensors. It's all about getting accurate, reliable data.
Right. Building that full, accurate picture.
Yeah.
Okay, so now things get interesting. You mentioned a puzzle.
Ah, yes. The puzzle. This involved a different file. S001R02.edf.event. The .event file.
Okay, an event file. Why is that so critical?
Because in EEG, event files are like the script or the timing cues. They mark exactly when specific things happened during the recording.
Like when a light flashed, or when the person was told to imagine moving their hand.
Precisely. Without these markers, you just have this continuous stream of brain activity, but you don't know what the person was doing or experiencing at any given moment. It's like watching a movie with no idea what the plot points are.
Okay, so you need the event file, but it was a puzzle.
It was. This specific .event file,
Yeah.
its format was a complete mystery. We couldn't find clear documentation for it anywhere for this particular dataset's events.
Uh-oh. So what do you do then? Data forensics.
Pretty much. We tried opening it as plain text, looking at the raw binary data, just hunting for patterns, any clues.
And did you find anything?
We found hints. There was definitely a readable text header at the very beginning, but then it just turned into what looked like, well, binary code.
Gibberish?
Sort of. We started seeing potential patterns, though, like maybe the last 8 bytes of each chunk represented a time and an event ID. It felt like cracking a code, but we didn't have the cipher key. It was pretty frustrating.
I can imagine. So could you actually extract the events using the standard tools?
Well, that was the real problem. We tried using a standard function in MNE-Python, MNE.events from annotations. It's designed for this, but it only found one single event type in the whole file. T0. Just T0. But your forensic work suggests that there should be more.
Exactly. That's what was so confusing. And it really highlights a common challenge in data science. Sometimes you get data, but without clear documentation for its specific format, interpreting it correctly is incredibly hard, like having a map in a language you don't speak.
Wow. A map with invisible ink, like you said before. You know there's more there, but you can't quite see it. So that leads us to epoching. The saga. The epoching saga, yes. So to analyze how the brain responds to things, you don't usually look at the whole continuous recording at once. You epoch it.
Epoching. Explain that again.
It's like taking scissors to that long stream of data. You cut out small, precise windows of brain activity centered around each event marker. So if T0 is our event, we cut out a little chunk of data right around time T0.
Okay, like taking snapshots around key moments
Yeah.
makes analysis easier.
Exactly. You could average these epochs, compare them. But our first attempt, using that single T0 event, well, it failed.
Failed high.
MNE just reported all epochs were dropped.
Yeah.
And the error was no data.
No data, even though you loaded the data.
Yeah, it sounds weird, but it meant we were asking for data from a time before the recording actually started. We'd set the start of our epoch window, tmin, to a minute of 0.2 seconds. So 0.2 seconds before T0.
Ah. So if T0 was right at the beginning, you were asking for data from before the recording even began.
Precisely. Asking for a photo from before the camera was on, like you said. A classic mistake, but easy to make.
Okay, so easy fix. Adjust the start time.
Step 1, yes. We adjusted tmin to 0, start exactly at the event. But then, new error. A value error this time related to the baseline.
Baseline.
Yeah, basically how you reference the signal level. The error message actually suggested a fix, set baseline. This means use just the single time point at the event as the baseline, which is fine, sometimes necessary. So we did that.
Okay, progress. Did it work then? Did you get your epoch?
Nope. Still dropped. The single epoch was still dropped.
Oh, come on. Why this time?
This time, the system gave us more detail in the drop log. It listed specific channels, SC5, C5, T7, others saying they exceeded a rejection threshold of 150 microvolts.
150 microvolt. Is that high? What does that mean?
It is quite high for brain signals. That threshold is there to automatically reject epochs contaminated by large artifacts, things that aren't brain activity, like eye blinks or jaw clenches or muscle tension. These create much bigger electrical signals.
So basically, even though you fixed the timing, the data in that specific window on those specific channels was just too noisy, likely due to muscle activity or something.
That's the idea. Our microphones picked up loud non-brain noise right at that T0 moment.
Man, that sounds incredibly frustrating. Fixing one thing, then another pops up. What was the breakthrough? How did you finally get an epoch?
It was a mix of frustration and determination. The detective mode kicks in. The breakthrough was a bit of a compromise, really. We decided to temporarily remove the reject parameter altogether.
You just turned off the noise rejection.
We did, just temporarily. It meant we were saying, okay, ignore the potential noise for now. Just let us see if we can create this epoch at all.
And
it worked. Finally. We successfully created that one epoch around T0. It was a real yes moment, even knowing we'd have to be careful about the data quality later. Just seeing something was the win.
I bet. After all that troubleshooting, finally getting that window of data. So what did you do with it? What did that first glimpse look like?
We visualized it, plotted the raw signals for that epoch. It was our very first proper look at the brain's electrical activity around T0, seeing those squiggly lines finally appear.
Yeah, that was satisfying. Puzzle piece found.
Okay, so Epoching conquered, at least for one event. You have data you can see. Now, the really fun part, bringing it to life, right? Making it visual.
Absolutely. But first, a quick sanity check. We did that channel check comparing the names from our WFDB calibration file against the standard 1005 montage map.
Did they match up? Mostly, yes. Lots of common channels, but there were some discrepancies, some differences in naming or maybe placement. Another real-world data thing. Standards are great, but data sets don't always follow them perfectly. You have to double-check.
Always keeping you on your toes.
Yeah.
Okay, channel mapping sorted, then the big visualization, the aha moment. You mentioned Topomap.
Topomaps, yes, they are fantastic. Think of them as 2D maps of the scalp.
Okay.
And colors spread across this map show where the electrical signal is strongest or weakest at one specific instant in time. It's like a heat map of brain activity. Really intuitive.
Cool, so you can see patterns, like maybe activity is strong over the motor area.
Exactly, you can see the spatial distribution, but we didn't stop at just static pictures.
Ah, right, you made it move, the animated brain.
We did. This is a really exciting part. We took a sequence of these top maps, calculated for successive time points within our epoch, and stitched them together into a GIF animation. We use the Imagery library in Python for that.
So instead of just one snapshot, you get a little movie.
Exactly, a dynamic view. You see the electrical activity changing, shifting, rippling across the scalp map over time. It really gives you a feel for the brain's dynamics. Watching those waves move across the head, it brings the data to life in a way static plots just can't.
That sounds amazing. Seriously, the ultimate payoff after all that detective work. From a misidentified zip file through error messages and weird formats to an actual moving picture of brain activity, that's incredible.
It really felt like it. Makes all the troubleshooting worthwhile. It's that satisfaction of turning complex, messy data into something understandable, even beautiful.
Okay, let's quickly recap this whole journey then. We went deep inside the brain, no surgery needed. We started by finding and downloading that raw EEG data from PhysioNet, the S001RR01.edf file, 64 channels strong.
Yep. Then we pre-processed it, filtering out noise between 1 and 40 hertz to focus on the key brain rhythms. And then we hit the event file mystery, S001R02.adf.event.
The infamous event file, our mysterious friend. It gave us that single teaser event, but stubbornly kept the rest of its secrets hidden because of that undocumented binary format.
Right, which led directly into the Epoching saga, wrestling with no data errors, then the rejection thresholds kicking out our data.
Until you finally made that breakthrough by temporarily removing the rejection criteria, just to create that one precious epoch around T0.
Our first window.
No first winner, exactly. And then the visualization finale, checking the channels, creating those insightful top maps, showing activity across the scalp.
And culminating in that fantastic GIF animation. Stitching the top maps together to watch the brain activity ripple and flow over time, the brain in motion.
From raw file to animated brain map.
It's an amazing process.
Yeah.
But that event file question mark still hangs there, doesn't it? That unsolved piece of the puzzle.
It really does. Without clear documentation for that specific binary format, we can't fully understand all the experimental events. We can't perfectly sync up the brain activity with precisely what the person was doing or imagining. It's a real hurdle. And it highlights something important, right? That data analysis, scientific discovery, it's often an ongoing process. Some puzzles just remain unsolved for now.
Exactly. Maybe waiting for a future deep dive or for someone to find that missing documentation.
So thinking about all this, what does it mean for you listening right now? Consider the incredible potential here. EEG is already used for diagnosing conditions like epilepsy for brain-computer interfaces.
Helping people control prosthetic limbs with thought alone. The applications are growing constantly.
So imagine, if we did have all the pieces to that event file puzzle for this data set and others like it, if we could fully decode these complex brain signals, perfectly aligning them with every thought, every action, every intention, just think. What hidden patterns, what truly profound insights into how our minds work do you think we might finally unlock?
Everything we do is available freely on bionichaos.com. Please consider supporting me on Patreon, and don't forget to click like, subscribe, and comment.
And there is a human in the loop as well. And yes, this dataset, we didn't have much luck with it. Well, because we couldn't actually decode the event files. And I have this time, a weird time resolution. Maybe we can try it one last time. Can we try decoding parsing, whatever. And let's see what we get. Probably would not get far. So probably be looking at a different data set next time. We're just unlucky with this one. Okay, you can run this. So using Colab, Google Colab. It's pretty good. So it's having trouble. It's changing the code. It's having trouble to access the file or something. It shouldn't be a problem.